So do biochemists measure the same kinds of concentrations inside synaptic vesicles that we saw in the Bruns and Jan experiment that we read on Friday? The answer is absolutely. These are numbers from biochemical extraction of transmitter out of purified vesicles from the torpedo electric organ, from more chromaffin cells, from dense core vesicles in pig platelets, in insuloma cells, and you can see very high concentrations not only of the transmitter itself, but also of nucleotides, ATP. So very, very comparable high concentrations of those and calcium. So very consistent across all kinds of secretory vesicles. So can we have more than one type of neurotransmitter in a vesicle? When I was in graduate school, there was something called Dale's principle, which said no, each neuron had a uniform phenotype and only had one kind of synaptic vesicle and one kind of neurotransmitter in it. We're now not so very sure about that. There's many instances where there is co-packaging of neurotransmitters. In this experiment, synaptic vesicles were purified from neurons in the midbrain of rodents. So we're looking at monoaminergic, perhaps even dopaminergic neurons, and the synaptic vesicles from them. And then they once they get the vesicles out, from the vesicle proteins, they do immunoprecipitations, and they're pulling down with a synaptic vesicle. Well, that's the whole synaptic vesicle prep. Synaptophysin, which is another prominent vesicular protein, and the glutamate transporter, the vesicular glutamate transporter, and the vesicular monoamine transporter, and then an indifferent protein. And from those pull downs, then they stain with an antibody to the vesicular monoamine transporter. And so the vesicular monoamine transporter recognizes itself, but it also shows that anything pulled down with antibodies to the vesicular glutamate transporter also has bound to it the vesicular monoamine transporter. And then they do the pull downs in reverse, and they probe for the vesicular glutamate transporter, and they find that vesicular monoamine transporter has vesicular glutamate transporter associated with it, suggesting that in the same population of vesicles, and perhaps even in single vesicles, these two different neurotransmitter vesicular transporters coexist. What would be the function of packaging two neurotransmitters together? Certainly there might be some postsynaptic responses that could be amplified or complementary, especially if the monoamine receptors had longer time responses or were setting some kind of a baseline within the tissue and had modulatory influence over the postsynaptic response to glutamate. But what about the contents of the vesicle itself? Could the glutamate transporter actually facilitate packaging of the monoamine? Do they help each other in terms of preparing the vesicle for its main job? So in this experiment, they're using tritiated serotonin uptake as the assay because it's the easiest monoamine, tritiated monoamine, to work with. And they first express VMAT2, the vesicular monoamine transporter, in the plasma membrane of HEK cells, and they look at the amount of serotonin that is taken up in the absence of anything to stimulate the glutamate transporter. So they're using aspartate, which is not taken up by the glutamate transporter. And in the presence of aspartate, they get a basal release or uptake of cargo on the vesicular monoamine transporter. And that does not change when they substitute glutamate for that aspartate. So there's, as long as the only transporter present is the monoamine transporter, then it doesn't matter which amino acid happens to be present in the extracellular space. However, if they co-express the vesicular monoamine transporter and the vesicular glutamate transporter, under those conditions, then when glutamate is present, they get an increased amount of monoamine or serotonin uptake, about 10 to 15% more. So it appears that the glutamate transporter, by bringing in more glutamate, is assisting in acidifying the interior space and therefore increasing the amount of monoamine that can be trapped inside here, the HEK cell, which is being used as a model vesicle. Similarly, they purify synaptic vesicles from rat brain using the endogenously expressed transporters, assuming now, because they've done the protein pull downs, they've got both. And they show that with aspartate and glutamate at normal chloride concentrations, when glutamate is present, they see an increased amount of serotonin uptake into synaptic vesicles, as predicted by the HEK experiment. And that is the same no matter what the chloride concentration is. So going to a higher chloride concentration does not change the ability of the glutamate transporter to bring in not only glutamate, but to acidify the vesicle more and increase the capacity for monoamine. So the co-packaging not only might have a postsynaptic response difference, but it can make a difference on the filling of the transmitters in the vesicles. And the co-packaging may actually be a modulation of presynaptic release as opposed to a modulation of a postsynaptic response.